Right, we take another example. So we can relate real life experience, real life things to learn and understand certain phenomena. Now, what will happen accidentally if you cut your finger? So your experience is the blood is going out of your body. Probably you will notice few drops coming out, and after that the many drops are coming, and these drops liquid maybe contaminate the surface or the floor like things, but after a while this place gets solid. That means the liquid becomes solid. Usually blood is, when it going out of our body, it becomes solid. Right, now if a sample is taken from our body, so usually if you notice, blood is mostly taken from the hand, this, right? We have learned vein and arteries. Arteries are very deeper. So arteries are like this. They are running very deeper. Why is that? Because they have a high pressure. Arteries have high pressure. If it is a damage to the artery, it's very difficult to control the bleeding. Veins. Veins are more on the skin level. So this is a vein. This is a artery. Then the needle insert into the vein. So it should go into the vein. And then after that, if the flanger of the syringe push backward, we can collect the blood. So this is one way of collecting blood. So we can also collect from the fingertips, different ways, but generally, this is the preferred way of collecting blood. Then this blood can be collected in two ways. What is the two ways? The method is same. If we collect this method and let's say then the blood transfer to a normal test tube. And here, second situation, it is also transferred to test tube. According to our experience, we should know after some times the liquid is no longer there, it gets a solid. The blood will get solid. Solid means not very hard, but it make a kind of a mass which is no longer liquid, which is no longer moving. Okay. Then if we leave the blood in a test tube, maybe 2 hours or 1 hour, we can notice it formed like this kind of a solid mass, and pale yellow color, a liquid. Pale yellow color liquid. Right now, what is important to understand, what is this liquid? What is this solid? This solid consists of cells, which include red blood cells and white blood cells. So this is no longer liquid. I would say it's a semi-solid. It's like a hard, quite hard material, not like a liquid, semi-solid. And usually we call this clot. A blood clot. A blood clot is used, the word used, blood clot. Right, now what is the word used for the liquid? We'll come to that later. Now in the second situation, this stay as a liquid. This stay as a liquid. Now you can understand, you know, blood transfusion. If a person is severely sick or lost a lot of blood, the blood is given to the person. This is called blood transfusion. In blood transfusion, there is a bag like this, and this bag containing the blood, and then the needle inserted into the vein, and slowly, maybe let's say 1 hour period, slowly blood is given to the person. That's called blood transfusion. Now, in this case, this stay as a liquid. Liquid, at least, let's say 2 hours. Now, what is your experience if you cut your finger or something happened, bleeding happened? How long will it take to stop? Maybe minute, 1 minute, 2 minutes. So after 1 or 2 minutes, it make a solid clot, solid mass. We call clot. Conclusion is, when blood is out of the body, within minutes, it becomes solid. Okay. Now, why then they stay as a liquid is a question here. Why they stay as a liquid? And here it clot, and here it's not clot. Okay, then we go further with this not liquid one. We'll try to find answer at the end why this is happening. And if we want to know the composition of the blood, we take, let's say 10 milliliter, 10 milliliter of blood, and we can take from a person with syringe, and then we can place in a test tube. After that, normal labs mean there are labs called diagnostic lab. What is diagnostic lab? You will see many places, they collect sample and they take to these hospitals. The hospital has diagnostic lab. So this is diagnostic mean their main focus is to find the disease of the person. The second type is a research lab. So they are not bothered about the disease diagnosis, they do research. And school lab, just for the learning purpose, school lab. So many places, there is an instrument called centrifuge. Now centrifuge, they have holes like this, and we can place the tubes. So we can place inside the holes. After that, we can close it, and then we can change the rotation speed. So this is the axis, and it is going to rotate around the axis in high speed. So sometimes it is 3,000 round per minute. 1 minute, it rotates 3,000 times. Sometimes some machines, within a minute, they rotate 20,000. Ordinary people, like general public, like things, we call it spin. We just use the word spin. But scientific word is centrifuge. So this is a normal instrument in the lab, used for many purposes, for analysis of blood, different chemicals, fluid separation. If there is a solute and liquid, it can separate, because when it rotates, molecules move through the gravity. So molecules move through the gravity. Then molecules, they have a heavy molecules, they can go faster. It is something like this. If you take a mixture of a sand, little stones, small stone and dust-like particles, and then you can put in a jam bottle, and then you shake it and you rotate it faster and leave it sometimes, the stones going down first. And the small things are in the middle, tiny things are on the top. That is a example of centrifugation. This is a method usually we do in a lab to separate particles based on the gravity. Now, if you do this, we can see this kind of a picture. After that, what we observe, a red color area, something like this. And there's a white color area in the middle, which is very thin. And after that, this much is a pale yellow color area. So we can see 3 layers. So these 3 layers, this is a liquid, this is the cells. So 2 types of cells, the middle, white blood cells. White blood cells are also called leukocyte. And this is a red blood cell. Red blood cells are also called erythrocyte. Right. Now you understand 1, 2, 3. There are 3 components in the blood. Liquid one is called plasma. Liquid part is called plasma. And white blood cells, they are not much. If you look, there's a tiny layer in the middle, and red blood cells. Generally, cells are 45%, plasma is 55%. But these values are changing, so I'm taking the average values, but that can be changed. 45% cells, 55% plasma. Right. Now we understand what are the main component of the blood. Liquid portion is called plasma. Cells, 2 types, leukocyte and erythrocytes, white blood cell and red blood cells. Now coming back, why, if we take blood and keep in the tube, it again separate without this centrifuge? We don't do any centrifuge like things. Just leave it sometimes, and there's a clot in the middle, in the bottom, and the liquid part is there, and the tiny middle layer there. Right now, Do we call this plasma now? So we are not going to use the word plasma. We use serum. Serum is a liquid part. Now we're going to understand why it is called serum here, why it is called plasma here. Now the major difference is I just label it A. This is A. This is B. So what happened A and B? A, blood clot form. B, clot not form. A, clot form. B, clot not form. Now again we have a question. We can understand blood clot. Blood is clotting when it is outside. That's a natural process. Because if there's a bleeding, blood clot. We don't understand now why it is not clot. Why it is not clot? They add chemicals not to clot. Here, when doctor is taking blood, most of the time, some chemicals are adding not to clot. And here, they add chemical not to clot. Okay, now further elaborate the thing. When we learning unit 3, we have learned leeches. We have learned one lesson. We have learned about leeches. Now, why leeches are important? Because we can understand a little bit of this if we understand this. Some of the insect like leeches, like annelids, they secrete chemicals which prevent blood clotting. Mosquitoes secrete chemicals which prevent blood clotting. Now, when it comes to mosquitoes-like things, you must be wondering certain diseases like mosquito-transmitted disease. So we know well some diseases transmitted by mosquitoes. Common disease we have in our country is dengue. So it's a virus. Now, how we can understand this? This is a skin, this is a blood vessel. Now, mosquito insert this, they are, what is called this, sting or the needle-like things in the mouth. And then, first thing is, chemical is going this direction. That means saliva is going this direction to the blood. And these chemicals prevent blood clotting. With that, disease-causing agent can transmit. Like virus, dengue virus is going with the saliva. When it inject the chemicals which prevent blood clotting. Otherwise, if they're taking the blood out, there's nothing coming into our body. They're taking things from our body. No, it is not the case. First, inject things to our body. That's a problem. We get these diseases. second, then they take the blood. So we add chemicals not to prevent the blood. So these leeches, like animal doing the same things, they secrete chemicals and it prevents blood clotting. Now here, the B option, they add chemicals not to clot the blood. So we haven't learned blood clotting yet, but why serum and plasma are different? Serum, what is left after clotting? Serum is what is left after clotting. Some of the proteins are not in the serum. Serum contain less proteins because which used to clot the blood. But plasma has these proteins. That's why serum and plasma are two different things. Plasma has clotting proteins. Serum has no clotting proteins, which is used. Naturally, when blood clot, this clotting protein used. Then serum has no clotting protein, plasma has clotting protein. And in this case, blood transfusion, they also have add the chemicals which prevent clotting. So they add chemicals which prevent clotting. Otherwise, after a few minutes, the tube is stuck and everything is clot, becomes solid, cannot transfuse the blood. And doctor wants to analyze the composition, like what is a plasma composition, cell composition like thing. Again, they have to add chemicals in order to prevent blood clotting. So we need to add most of the time chemicals to prevent blood clotting for analysis. So basic composition is 55% of plasma, 45% of red blood cells and white blood cells. Okay, do you have any question about this blood, main composition of blood? Okay. Right, if you don't have a question at the moment, try to remember at least these facts very clearly. Composition, main composition of blood, 55% of plasma, 45% of red blood cell and white blood cells. And what is the difference between serum and plasma? Difference is serum use clotting proteins because it clot. And plasma has all the clotting proteins because it's not used up. In this case, this tube, no clot. Only clot is in this tube A. Right, next, we are coming to another fact. There is another type of cells. We will try to come to that one. Now, when we are learning white blood cells or erythrocytes, white blood cell or leukocyte, right here, we're going to leukocyte or white blood cells. Now we have a complete lesson coming up called immunity. It's called immunity. Now, when we're considering immunity, and you can understand there is a complete lesson for immunity, the main component of the immune system is the leukocytes, white blood cells. So it represents our main immune component. So they are cells. And when you're going to learn immune system, you're going to learn different type of cells, white blood cells. I think it is better you have a little bit of idea what type of white blood cells in the blood. So get a little bit idea type of white blood cell in the blood, because we need it to understand immunity. Okay, in short, there is a cells in this category. These cells are very special. These cells have a special nucleus. The nucleus is like this. It divides into compartment. And there is a small fluid fields, vesicles, we call them granules. So we have learned vesicles, they are membrane bound, fluid filled small particle in the cells, and granules are the same things. They have a different fluid inside. And there is another cell which has again a nucleus divided into two, and they have a different type of granules, different colors. And there's another one. So they are again a little bit difficult to see this nuclear division into two, and again they have granules. Okay, additional question. Should we write the answers exactly which is in the resource book? I think it's better that I will show you a example. My answer is yes. My answer is yes. I have done this previously. I don't know who asked this question. Okay, I showed you previously, taking one example, the essay type of question. I took the marking scheme and I took the question, I took the resource book. So, I mean, you can also do it, right? Take one question, take the marking scheme, take the resource book. Now you can match how many points in the marking scheme which is matching with the resource book, and do for another question. At least you do for 3-4 questions. It's better that you understand yourself rather someone is telling that it is like this, like this. Take 3 essay type question, take the marking scheme, and take the resource book. And then you highlight marking scheme point with the resource book points. After that, you came conclusion. The marking scheme point, there are 36 marking scheme points. Out of that, resource book has exactly 36, or what is your result? 35 or 20 or 15. You just get it values. Then you realize as a percentage, how many points written in the resource book. My answer is again, you have to write answers according to the resource book. Now, this is again, this question, should we write the answers exactly which is in the resource book? Yes, it's a good question. It's a very good question. I'm also a little bit of stuck because I don't like if someone is learning, his learning power is only memorizing, the person is not intelligent. The person is not intelligent. Any person who can memorize, then he can repeat the same thing, he can get the marks. But unfortunately, your exam is like this. I have this, I think, I clearly showed you in Unit 2. I took one example. I remember it, recent new syllabus, enzymes. Right. I can, I mean, you know, you have joined later. I don't know whether I have this recording, but I can give the document, like I can WhatsApp the document. It's unfortunately it's like this. For example, one marking scheme point come under the enzyme. You can write enzymes are proteins. Enzymes are proteins, full stop. But marking scheme point is enzymes are mostly protein, which is in the resource book, which is in the marking scheme. So then your answer question, your question answer is yes. Unfortunately, yes. That means if you miss mostly, will I get a mark? No. This is a very strong message, but majority of the people, student, they don't understand this fact. You have to write according to the resource book, exactly in the sense, it's mostly like we have information in our brain, but this information not true information. Let's say photosynthesis. Any lesson you take, okay, light dependent reaction, you can write. You can write light dependent reaction. But if you write it and check with the resource book, I think it's matched mostly with 99% it's matching. Maybe one or two word missed. It's not according to that. Then it's okay. So then it is not a big issue that you don't make a fear. You don't, exactly, I had to remember each and every word. No, it is not like that. We try to remember the truth. We try to remember the facts. These facts and truth is same everywhere. For example, let's say if you ask, let's say structured question, if you ask, if you ask a question, okay, we take a question, we make a question, write the difference between serum and plasma. You have a question to answer in the structured question, write the difference between serum and plasma. Then you write, serum has no clotting factors. Serum has no clotting factors. And then you can write, serum has no clotting factors, whereas plasma has clotting factors. You get the marks. Now you go and check the resource book, what is serum? So you can check the resource book, what is serum? Now I give this message, don't make any fear that you have to really remember each and every word as in resource book when you're writing answers. So large amount of information we are learning can be used, but there are certain cases we have to use the right word. We have to use the right word to get the marks. That is certain percentage, not the major percentage. Certain percentage are like that. As a result, if you smoothly go on and try to remember things, it is okay. But later, you put additional effort to remember certain things as it is. As I told you, you can win this game in one way. We need to finish the syllabus as soon as possible. Then you spend many times for memorizing, because understanding component is done. You understand well, then it's easy to remember. Why is that? The real life experience of me, now 2023 student, sometimes call, so I went to that place, I don't get it, can we start class like this? I mean, why you have waited so long? So if the syllabus is still 2024 student, 2023 student, they are doing now unit 5. So how many months for the exam now? Truth is, most of the students lost at the last part. Last 10 months, they lost. It's too late after that. This is not just like a cricket match. At the last overs, you can hit the sixes and win. You can't do that. We are playing with the brain. From the beginning,